All right, so I want to talk to you a little bit about this model of CFTR and how you might be able to best use it for your infographics. So I do expect to see this on your infographics, so you're using a part of it to make your model, but it's important to provide the context of what area you've selected out of this full protein to print. Now, we're not printing the whole thing for each of you, so you've isolated that section, you've given me that file, I'm printing them now. But what you should do is also provide a context of where it is in this model. So let me clean this up a little bit. Um, and um, and what we have is this model, right? So it is what you started from and what you worked with, and so you would restrict that down. But one thing you could do is identify a region. So let's say that my mutation that I wanted to select exists in this transmembrane alpha helix. So what I might do is select, well, let me pick this one and this one. All right, so I might just, if I've selected all and color it white, or maybe you color it a different color with a different background, because you need to think about your infographic. You probably don't want to print a black background of your photo. So maybe background white, and you want to color this light gray, or something, right? So you still have that shadow, but you may just want to select 1021 through 1058 and color that light blue, or color it, I don't know, sorry about that, that's Ari in the background there, orange, right? So you know that your print is of this region right here, and you may even want to put your mutation on it. So I'm just making up a mutation. I have not verified this. This is just for example's sake. So if I select 1035 and sidechain and do a space fill and color it CPK, so this may be what I print, what's here in orange. I've printed out for you to use, but it also provides you the context of where this is in this full protein, right? So that's one thing that I hope to see, and all you would do is hit this camera, save it, insert that into your infographic. So that's definitely one thing. But let me show you something else that I've done. What I've done is gone through and color coded. And now it's a funky color code system, I acknowledge that. Each of these transmembrane sections is a different color. There are 12 of them all together. Here's the this reddish crimson color is the regulatory domain, and then the purple and the yellow are the two sides of the nucleotide binding domain. And I really haven't shown you anything else besides that, so that's color coding, and that may be something that you want to show in your model as well. So you may want to start from that structural file, CFTR structural model PDB file, and do something like this to help show the different regions of the protein and how you went about selecting the different components. And it's actually kind of turned upside down, because in the cell, the way that I drew it in class the other day would look like this. This inside the cell, and this would be in the membrane, and the outside the cell above the protein altogether. Here's yet another model that I thought was really interesting, because what I did was went through those papers that we prepped for class the other day and highlighted a few important structures here. So we talked about how this CFTR protein only allows chloride ions through, and chloride has that negative charge. So how in the world does it repel positive charges? Well, if you look inside the channel, right here, I've turned on arginine, which is located right here, arginine 352, which is what was suspected in the first paper that we read. This is what one of the predictions was, because this has a positive charge, it's mostly nitrogenous, with one central carbon in that side chain, it has a positive charge, and that positive charge is going to repel other positively charged ions or cations, and it's going to draw in anions. Now, anions are going to be drawn into this space, but only when this space changes to this right shape and size will chloride ions be allowed through. And that's really what brings me to these other things that are labeled or shown on this side over here. So let's look at the top example right here. So what I have here is 2 amino acids and then a third one with one in between. And this third one is a serine. And these two right here are 2 basic sites. So this is called a diabasic site. This is where the phosphorylation happens. So the first part to regulate this protein requires phosphorylation, and then a secondary ATP comes in, and that changes the transformation of the pore to allow chloride through. And so this is one of those sites, but there's actually a lot of redundancy in this protein, which is really important to have that redundancy, because if there was a mutation on one of these sites, another site could do its job, which is really good for your own survival, to inherit all that redundancy. So you're going to see that same motif over and over here. So we've got the serine, an open space, and we've got 2 basic side chains. We're going to have that over here as well. So we've got serine, basic, basic, serine, basic, basic, serine, basic, basic, serine, basic, basic. And what I've done is just highlighted those so you could see those. And this could be a model that you want to show. All I did was use some of the resources from the papers and a map, one of those gene maps that you use for insulin. I've got one that's got some of these phosphorylation sites or diabasic sites labeled, and I'm happy to share that with you in class before the presentation. So you could add this as well. And you could use these files with a computer during the presentations for JMOL, but I don't have a computer to provide to you on the day of the presentation. So if your team has one, you may use it. The other thing that I've done also in this model is shown right here is phenylalanine 508. So this is that common deletion. And again, that deletion is not in the amino acid. It's the amino acid's missing because the genetic material, the DNA for it, was deleted. And because it was deleted, this phenylalanine would not appear in those most common recessive alleles of cystic fibrosis. Okay, so there's a lot there that I've shown, and lots of different ways of showing it, right? So you could take this, and you could also say, if you want to do another model of select hydrophobic, oh, well, there's a code. I'll find it. You can find hydrophilic or hydrophobic. You could actually look for it too. And you could highlight them as with different color coding. So you could see, well, because these transmembranes have a higher proportion of hydrophobicity than the nucleotide binding domain, that these are going to be embedded in the cell membrane. All of those things are important to think about, important to show. It depends on what you think is important and what's important for you to evaluate. Now remember that your grade, the content grade, is coming exclusively from the video and your infographic. But the skills, 30% of the grade for this project is on presentation day. And what that means is that you, even though the team produces the same thing, you may not have the same grade as other people in your team, and that really depends on what you've contributed and how much you know and how you can answer and have a conversation with our guests that will be coming in. Okay, so I'm going to stop there. I'm just shy of 10 minutes, and that sounds good to me. So good luck, and work on your infographic today.